In this video, we're going to cover photosynthesis, which is the process by which plants use sunlight, water, and carbon dioxide to produce oxygen and energy in the form of sugar. Before we break down the process of photosynthesis, let's first look at the ecological importance of photosynthesis. Because, like I said in the cellular respiration lecture, there's no point in going through the steps and trying to memorize them if we can't see the big picture and see the beauty of this process. So, why is this important? Because almost all life on Earth is sustained by photosynthesis, either directly or indirectly. All organisms, including humans, need a constant supply of energy to allow the millions of reactions that are occurring in each cell to occur, because these reactions are what help them survive, grow, and reproduce, as well as maintain their structure and metabolic needs. Let's break this down further. Let's take a look at how photosynthesis connects with cellular respiration and how we can classify organisms according to their energy and carbon sources. Why carbon? Because the organic molecules that make up cells are built on the basis of carbon, and cells also use carbon-containing compounds. All right? Organisms that capture energy from the sun are called phototrophs. Think photo equals photosynthesis. And organisms that obtain their energy from chemical compounds are called chemotrophs, and animals are examples of chemotrophs. But the energy from these chemical compounds is fundamentally attained from the sun. Can you see how almost all life on Earth depends on photosynthesis? Now, when it comes to where organisms get their carbon, organisms that are able to convert carbon dioxide, which is an inorganic form of carbon, into glucose, which is an organic form of carbon, are called autotrophs or self-feeders because they can do it themselves. Plants are photoautotrophs because they get their energy from the sun and they can automatically convert inorganic forms of carbon into organic forms. They are able to produce their own food using light energy. They are self-feeders. They can feed themselves. They're independent organisms that need no one else besides the sun. Then we have organisms that don't have this ability, and instead, they get their carbon from organic molecules synthesized by other organisms. These organisms are called heterotrophs, or other feeders. Humans and organisms and other organisms are heterotrophs. Animals are chemoheterotrophs, okay? Think chemo equals energy from chemical compounds and hetero equals other feeders. All right, that's how we can classify organisms according to their energy source and carbon source. Another important function of photosynthesis is that it has an impact on the composition of the Earth's atmosphere. If we look at our equation here, oxygen is released as a byproduct. Without this process, oxygen requiring life would not survive and there would be no oxygen source and you wouldn't be here, okay? And one more thing, large amounts of carbon dioxide are also removed from the atmosphere by photosynthetic organisms who then use the carbon atoms to produce organic molecules to feed themselves. Without photosynthetic organisms, carbon dioxide would accumulate in the atmosphere. Now, with the glucose molecule here that is produced from photosynthesis, it gives organisms two essential resources, energy and carbon. Okay, how does this connect with cellular respiration then? Glucose is a major source of fuel for cells. We can harvest the chemical energy through processes like cellular respiration and fermentation, which produce ATP or adenosine triphosphate. We can release energy for the cells to use. It's used as a fuel for cellular respiration. The fuel is broken down using oxygen to make ATP. And the other products of cellular respiration, carbon dioxide and water, are used in photosynthesis. That's how these two processes connect. Let's now subtract complexity and break down where photosynthesis occurs. In most plants, the leaves are the major sites of photosynthesis, specifically the chloroplast. This is the organelle that absorbs light energy and uses it to drive photosynthesis. Let's zoom in. Chloroplasts are found mainly in the cells of the mesophyll. The middle layer is the interior of the leaf. There are also small pores here called stomata, singular stoma. And these pores allow carbon dioxide to diffuse into the mesophyll layer and oxygen out. And when it comes to water delivery, veins carry the water that the roots have absorbed to the leaves. And veins are also used by leaves to carry sugar to the plant's roots and other non-photosynthetic parts. Now, each mesophyll cell contains chloroplasts, about 30 to 40. Let's go through the structure. A chloroplast, look at this masterpiece, has two membranes surrounding this fluid-filled space called the stroma. Inside the chloroplast is a membrane system in the shape of flattened link sacs known as thylakoids. They are stacked like cookies or poker chips, and each stack is termed a granum. The membrane of each thylakoid contains green-colored pigments called chlorophylls that absorb light. This is the green pigment that gives leaves their color, and the light energy absorbed is what drives the synthesis of organic molecules. Looking at this, the membranes divide the chloroplast into compartments: the intermembrane space, the stroma, and the thylakoid space. This organization allows the chloroplast to convert light energy to chemical energy. Different reactions occur in the different parts of the chloroplast. We can divide photosynthesis into two stages: the light-dependent reactions, this is the photo part of photosynthesis, and the Calvin cycle, this is the synthesis part. Let's go through the light-dependent reactions. The light-dependent reactions take place in the thylakoid membrane. This is where, if you recall, chlorophyll and the enzymes involved are located. At this stage, light energy is absorbed by chlorophyll, which is converted into the chemical energy of ATP and the reduced electron carrier, NADPH. The two molecules are needed for the next stage of photosynthesis, the Calvin cycle. But before we look at this in greater detail, let's look at light energy, because photosynthetic organisms only use a specific component of sunlight. What is light energy? Light is a form of energy known as electromagnetic radiation or electromagnetic energy, and the sun emits a significant amount of electromagnetic radiation or solar energy. Humans can only see a fraction of this energy, which is referred to as visible light. We can describe the manner in which electromagnetic energy travels as waves, okay? This is an energy wave, and we can determine the amount of energy of a wave by measuring its wavelength, the distance between two consecutive similar points in a series of waves, such as from crest to crest or trough to trough. Okay? And each wavelength corresponds to a different amount of energy carried. The longer the wavelength, or the more stretched out it appears, the less energy is carried, and the short, tight waves carry the most energy. These range from less than a nanometer for gamma rays to more than a kilometer for radio waves. This entire range of radiation is known as the electromagnetic spectrum, and the sun radiates the full spectrum of electromagnetic energy, but the atmosphere acts like a selective window, allowing visible light to pass through. Visible light is the radiation that drives photosynthesis. Okay? Now, when light meets matter, it may be reflected, transmitted, or absorbed. And substances that absorb visible light are known as pigments. They are light-absorbing molecules. There are several types of pigments, and each absorbs only certain wavelengths or colors of visible light. Pigments reflect the color of the wavelengths that they can't absorb. So for example, if a pigment absorbs all wavelengths of visible light, it appears black. That's why when we look at a leaf, it appears green, because chlorophyll absorbs violet, blue, and red light while reflecting and transmitting green light. Because green is reflected, chlorophyll appears green. All right? Now, there are three types of pigments in chloroplasts. Chlorophyll A, which is the key light capturing pigment that participates directly in the light reactions. Chlorophyll B, which is the accessory pigment, and the carotenoids. Each type of pigment can be identified by the specific pattern of wavelength it absorbs from visible light, which is its absorption spectrum. Chlorophyll absorbs violet, blue, and red light, so these colors work best for photosynthesis, while green light is the least effective, okay? And this leads us to how the light-dependent reactions actually work. Let's go through this. Photosynthetic pigments, such as those we mentioned before, are light-harvesting molecules found in the thylakoid membranes of chloroplasts. They are organized, along with their associated proteins, into complexes called photosystems. So our photosystem is located in the membrane of thylakoids. So this right here is a photosystem. Each photosystem is made up of a reaction center complex surrounded by many light-harvesting complexes. This right here contains proteins and hundreds of chlorophylls as well as other pigments. Let's break down what happens. A pigment molecule in the photosystem absorbs one photon, a quantity or packet of light energy at a time. When a pigment absorbs a photon, it's boosted to a higher energy orbital and it's said to be in an excited state. Let's slow down for a second and break this down further. Electrons are found in different electron shells. The 3D space where an electron is found most of the time is called an orbital. Electrons vary in the amount of energy they have. Remember, there's kinetic energy and potential energy, which is stored energy that matter has because of its location. Now, electrons have potential energy because of their distance from the nucleus. The potential energy of an electron is determined by its energy level. The farther it is, the greater its potential energy. And electrons close to the nucleus have less energy. As it moves from one shell to another, it either absorbs energy or loses energy. Okay, so bringing this back to a pigment molecule, it's considered to be in its ground state when the electron is found in its normal orbital, right here, the shell close to the nucleus. And when light is absorbed, this will push an electron to an orbital of higher energy. This electron is in an excited state. It's got spikes to show this, okay? And when one of these pigments is excited by light, it's going to transfer energy to another pigment, and then to another pigment. And they're going to keep transferring energy towards the central part of the photosystem called the reaction center complex. The reaction center complex is an ordered group of proteins that contains a pair of chlorophyll A molecules. This is known as the special pair. Once energy reaches the special pair of chlorophyll A molecules, the photon causes an electron in the chlorophyll to become excited, and it's going to pass the electron to another molecule called the primary electron acceptor, where it will get passed on through an electron transport chain, which we'll break down in a minute, because there are actually two photosystems in the light reactions that work together. Let's break this down first. There's Photosystem II and Photosystem I. Photosystem II comes first, but it's named as second because it was discovered after PS1. I know, they just had to confuse us even more and make this even more difficult for us. I feel you, but this will be a walk in the park. Let's look at the key difference between the PS2 and PS1. With the special pairs, the chlorophyll A special pairs of the two photosystems absorbs light at different wavelengths. PS2, the PS2 special pair, absorbs light with a wavelength of 680 nanometers in the red part of the spectrum, which is why it's called P680. Whereas PS1 special absorbs best at 700 nanometers, and it's called P700 in the far red part of the spectrum. Another difference is that once an electron is lost, the PS2 reaction center gets electrons from water, while PS1 reaction center gets them from the flow of electrons down an electron transport chain from PS2, which you'll see. Okay, so now that we've established the photosystems and the key difference between the two, let's go through the flow of electrons, a process called linear electron flow. And let's take a look at how we generate ATP and NADPH. So we have the two photosystems. The PS2 has the P680 special pair and the PS1 has the P700 special pair, a pair of chlorophyll A molecules. We'll label the other structures as we move forward. It'll make more sense. Starting with Photosystem II, when the P680 special pair absorbs energy, the electron is boosted to a higher energy state, which then transfers its excited electron to the primary electron acceptor. The electron will be passed on to PS1, Photosystem I, via an electron transport chain, which consists of the electron carrier plastoquinone, PQ for short, a cytochrome complex, and a protein called plastocyanin, PC for short. We'll come back to this though. Now, after the chlorophyll A molecule gives up its electron, it has a positive charge and needs a new electron. The electron in chlorophyll must be replaced. This electron comes from the splitting of water molecules. A water molecule is split into two electrons, two hydrogen ions, and an oxygen atom. The electrons are transferred to the P680 special pair. We can replace two donated electrons, all right? And the protons we produce, where does it go? It's released into the thylakoid space, okay? And the oxygen atom joins with an oxygen atom generated by the splitting of another water molecule, and we produce O2, oxygen gas. Now, let's go back to the electron transport chain. When an electron leaves PS2, it's transferred to plastoquinone, okay, PQ, then to a cytochrome complex, and finally to a protein called plastocyanin. If you've seen the cellular respiration lecture, this process is quite similar. As the electrons move through this electron transport chain here, it goes from a higher energy to a lower energy, and free energy is released. Some of this free energy is used to pump protons, okay, hydrogen ions, from the stroma outside of the thylakoid into the thylakoid interior, this thylakoid space. And the concentration of hydrogen ions in this space increases. This transfer of hydrogen ions, along with the release of hydrogen ions from the splitting of water, forms a proton gradient that will be used to make ATP, a process called chemiosmosis. We'll go through this in a minute, okay? We'll come back to this. I want to take a look at PS1 first. Similar to PS2, light energy is also transferred via light harvesting complex pigments towards the PS1 reaction center complex. And as a result, an electron of P700 is excited, and this electron is transferred to PS1's primary electron acceptor. And now P700 can act as an electron acceptor. We've donated the electron, so now we need to replenish the electron that we've donated. P700 is going to accept an electron that reaches the bottom of the electron transport chain from PS2. Recall when we talked about the key difference between PS2 and PS1? When PS2 loses an electron, it's replaced by the splitting of water, okay? Whereas PS1 gets them from the flow of electrons from PS2. Alright, then what happens? Where do we go from here? The photoexcited electrons are then passed from the primary electron acceptor of Photosystem I down a second electron transport chain through the protein ferredoxin, FD. Take note though that this chain doesn't generate a proton gradient, and so it doesn't produce ATP. The electron is then transferred to an enzyme called NADP plus reductase. This right here, NADP plus reductase. This enzyme transfers electrons to the electron carrier NADP+, and we make NADPH. The hydrogen ion is from the stroma. Then NADPH will travel to the Calvin cycle, where its electrons are used to build sugars from carbon dioxide. Okay? The other product needed by the Calvin cycle is ATP. So let's go back and let's go through how ATP is produced. Like we touched on before, the concentration of hydrogen ion increases inside the thylakoid space or interior, and there's a concentration gradient, okay? So we have a high concentration of hydrogen ions in the thylakoid space. So protons want to diffuse back down their concentration gradient from an area of high concentration to an area of low concentration in the stroma. And this is where the enzyme ATP synthase comes in. What ATP synthase does is it allows protons or hydrogen ions to flow from the thylakoid interior and into the stroma, and this stimulates the production of ATP. ADP and inorganic phosphate combine to make ATP. This process is called chemiosmosis. We're using energy stored in a chemical gradient to make ATP. Then this ATP is used in the Calvin cycle. Okay. Before we continue, let's quickly recap what we've just covered because that was quite intense. Let's sum it all up. Okay, what do? What's our first move here? First, energy from light excites an electron from chlorophyll, which is then transferred to the primary electron acceptor in Photosystem II, PS2. Because P680 lost an electron, it's replenished by the splitting of water. Okay, in the thylakoid space, water is split into electrons, which are transferred to chlorophyll. We also release hydrogen ions in the thylakoid space, and we also form oxygen gas. Now, the flow of electrons pumps protons into the thylakoid space, and this increases the concentration of hydrogen ions. In chemiosmosis, hydrogen ions diffuse down the gradient through ATP synthase and we produce ATP. Then when the electrons from the electron transport chain reach Photosystem I, they pass it to ferredoxin, which transfers it to NADP plus reductase, and we produce NADPH. Alright? And these two products then go to the Calvin cycle. They're used in the Calvin cycle. But before we break down the Calvin cycle, there's another path photoexcited electrons can take, which is called cyclic electron flow, which uses Photosystem I, but not Photosystem II. Recall that the path that we just covered is called linear electron flow, because electrons travel in a line from water through PS2 and PS1 to NADPH, okay? Let's briefly go through the cyclic electron flow. After leaving the PS1, the electrons are transferred to ferredoxin. And from ferredoxin, it goes to the cytochrome complex, then via a plastocyanin molecule to a P700 chlorophyll in the PS1 reaction center. Okay, so it's a cycle. This cyclic flow generates ATP because we're pumping protons, but it doesn't make NADPH since electrons are routed away from NADP plus reductase. And we also don't release oxygen in this process because recall the reaction takes place in Photosystem II. But why does this pathway exist? Why am I mentioning this? Cyclic electron flow may be prevalent in photosynthetic cell types with particularly high ATP requirements, or in some cases when the ratio of NADPH to NADP plus is too high, when there's insufficient NADP plus available to receive electrons, okay? So chloroplasts appear to switch from linear to cyclic electron flow. Okay. That's how light energy is turned into chemical energy stored in ATP and NADPH. And we also produce oxygen as a byproduct. Let's now move on to the Calvin cycle and break down how this cycle uses the products of the light reactions to synthesize sugar from carbon dioxide. Because now we have the fuel, we have the energy needed to build food in the form of carbohydrate molecules. These molecules are built on carbon backbone. So where does carbon come from? Carbon dioxide, okay? The gas that animals exhale. Do you know the carbon atoms of carbon in your body, in our bodies, were once part of carbon dioxide molecules in the air? Pretty cool, right? In plants, carbon dioxide enters the chloroplast to the stomata and diffuses into the stroma of the chloroplast, which is the site of the Calvin cycle reactions where sugar is synthesized. The cycle takes place in the stroma, the inner space of chloroplasts. Now, we can divide the Calvin cycle reactions into three main stages: carbon fixation, reduction, and regeneration. Let's go through carbon fixation, which is the initial incorporation of carbon dioxide into organic material. Okay, 3 carbon dioxide molecules are going to enter this cycle one at a time and combine with a 5 carbon molecule called ribulose bisphosphate. The enzyme that catalyzes this step is rubisco, and we produce a 6 carbon molecule or intermediate that is immediately split into 2 3 carbon molecules called 3-phosphorglycerate, 3-PGA. That's because this 6 carbon molecule is unstable, and so it immediately splits into 2 molecules of 3-phosphorglycerate. Alright, for each carbon dioxide molecule that enters the Calvin cycle, 2 3-PGA molecules are produced. So we have 6 3-phosphorglycerate molecules. Okay, quick recap here on the left before we move on to the next phase. Ribulose combines with a carbon dioxide molecule catalyzed by rubisco, and we produce a 6 carbon molecule that immediately splits into 2 molecules of 3-phosphorglycerate. That's the first phase. Let's move on to phase 2, reduction. This phase requires ATP and NADPH to convert 3-phosphorglycerate from the previous phase into a 3 carbon sugar. This process occurs in 2 steps. Let's break down the first step. First, each molecule of 3-phosphorglycerate is converted into 1,3-bisphosphorglycerate. Each molecule gets a phosphate group from ATP, and we produce ADP. We need 6 ATP molecules here because we have 6 3-phosphorglycerate molecules. Then the 1,3-bisphosphorglycerate molecules are reduced, okay? This is where NADPH comes in. This is the second step. NADPH is going to donate a pair of electrons to 1,3-bisphosphorglycerate, which also loses a phosphate group in the process and becomes glyceraldehyde 3-phosphate, G3P. Now, Where do ADP and NADP plus go? Well, they are needed by the light-dependent reactions. It's a give-and-take cycle. The substrates the light-dependent reactions produce are needed by the Calvin cycle, and the substrates the Calvin cycle produces are needed by the light reactions. It's a win-win situation. Drawing your attention to glyceraldehyde 3-phosphate, G3P sounds familiar. Do you remember where? G3P is a sugar. It's the same 3-carbon sugar that is formed in glycolysis, the last step of the preparatory phase. So one G3P molecule will exit the cycle to make glucose. This is our output of the Calvin cycle. Remember that 3 carbon dioxide molecules enter the cycle, so we produce 6 glyceraldehyde 3-phosphate molecules, and 1 can happily exit the cycle and be used to make glucose, while the other 5 must be recycled to regenerate 3 molecules of the ribulose bisphosphate acceptor. Alright? A quick recap before we move to phase 3. 3-phosphoroglycerate becomes 1,3-bisphosphoroglycerate. ATP is the phosphate donor here, and then NADPH reduces 1,3-bisphosphoroglycerate, which also loses a phosphate group in the process, and we produce G3P, glyceraldehyde 3-phosphate, which is a sugar, the same 3-carbon sugar formed in glycolysis. Out of the 6 glyceraldehyde 3-phosphate molecules the cycle has generated, 1 G3P will exit the cycle and go towards making glucose, while the other 5 are recycled. And this leads us to the next phase, the third phase, which is the regeneration of the carbon dioxide acceptor, ribulose. In this phase, we need 3 molecules of ATP. So now we have ribulose molecules ready to accept carbon dioxide again, and the cycle continues. Okay? That's the Calvin cycle. Let's summarize what we've just covered. The cycle needs to turn 3 times to make 1 glyceraldehyde 3-phosphate molecule that can exit the cycle and make glucose. In the 3 turns of the cycle, 3 carbon dioxide combines with 3 ribulose molecules, making 6 molecules of glyceraldehyde 3-phosphate. Okay? Remember that the carbon dioxide molecules enter one at a time. 1 glyceraldehyde 3-phosphate exits the cycle and is used to make glucose, while the other 5 glyceraldehyde 3-phosphate molecules are recycled, regenerating 3 ribulose acceptor molecules. Then with the ATP molecules, 9 ATP molecules are converted to 9 ADP, 6 during the fixation step and 3 during the regeneration step. And we also converted 6 NADPH to 6 NADP plus during the reduction step. And going back to G3P, a G3P molecule contains 3 fixed carbon atoms, so it would take 2 G3Ps to build a 6-carbon glucose molecule. So to make 1 molecule of glucose, we need 6 turns of the cycle. Okay? Wow, look at that. Before we end this lecture, let's take everything we've covered and sum it all up. Let's combine the light-dependent reactions and the Calvin cycle. So photosynthesis occurs in the chloroplast, and again, we can divide it in 2 phases, light-dependent reactions and the Calvin cycle. The light reactions occur in the thylakoid membranes, where we convert light energy to the chemical energy of ATP and NADPH. And we also split water and release oxygen gas. The ATP and NADPH produced during the light reactions are used to power the Calvin cycle, which occurs in the stroma. And we use ATP and NADPH to convert carbon dioxide to the sugar glyceraldehyde 3-phosphate. And we produce the substrates ADP, inorganic phosphate, and NADP plus as our byproducts, which are returned to the light reactions, and the cycle continues. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video, or you can view the entire playlist. Hey, stop procrastinating!